Aloha, everybody. My name is Zhiqiang Cheng, and I'm an associate professor at UH-CTAHR in the College of Tropical Agriculture and Human Resources, CTAHR. So today I'm gonna just do a brief update on field trials of systemic and biological fungicides for the management of coffee leaf rust. This is a part of my USDA SCRI project in collaboration with Suzanne and also USDA PBAR folks and other Puerto Rico collaborators as well. So I have my graduate student Lily, who is the co-author of this talk, and also Julia, who is the R4 coordinator here as well. All right, so we do not necessarily have to talk about the background of coffee leaf rust too much anymore, because by now everybody pretty much knows that coffee leaf rust has been here since October 2020. And these are the symptoms, and I think a lot of seminars and webinars, including Andrea's website, that you can see all the details. This is on the left picture. You see the early stage of the coffee leaf rust infestation, and on the right side you see the heavy infestation stage already. And so my update is only on the SCI project, and Julie has provided a separate webinar on our R4 coffee leaf rust trials, and depending on when you view her seminar or my seminar, you might have some background information on that as well. But I put this slide here. So this is our R4 team. We have Julie and James, and also Ning started with us last year, and also myself as a PI for the R4 program. So I will not go into details with R4 trials, but let's go straight into the SCI project on the coffee leaf rust control. So here you see Elizabeth, who is actually the student who is managing the research project, and she goes to Kona every so often to set up the trial and collect the data. And basically we are testing several systemic fungicides and also 2 biological control products for this project. And this project, I should just go ahead and minimize this, I guess. All right, put this on this side. All right, so these trials, I want to make it clear that there's no overlap between the SCI project and the the R4 trials. Both are actually going on concurrently at Kona Hills right now for the second year. It was the second year for the R4 trial, but the first year for the SCI trial. But for the SCI trial, we will repeat that for the second year, basically this year, 2023 to early spring 2024. That would be the second year of SCI project. So today I will only summarize the first year result from SCI project. So for this one, it was a fully properly designed research project. I want to briefly introduce the experiment design so that you guys can have some idea on how we run the trial. So we have essentially 5 treatments, including 3 systemic fungicides and 2 biocontrol products and 1 untreated control. That's why you see the 6 treatments here. We have 3 replications organized in a randomized complete block design. So what you see here is the 3 blocks: 1 block, 2 the second block, and the third block. And within each block, we have these 6 treatments randomized within each block. And within each treatment plot, we have 6 coffee trees, and when we do the evaluation, we only take the data from the inner 4 trees to kind of avoid any potential cross-contamination from the adjacent plots. So you consider the 2 bordering trees as kind of like the buffer area for each of these treatment plots. And here are the details of all the 5 treatments, the 3 systemic fungicides, and also the 2 biocontrol products. We don't necessarily have to go into the details of everything, but if you want to see the details, the details are here. Essentially, the active ingredient and the rate we use following the manufacturer recommendation, and also how many times we applied, also following the manufacturer recommendation at a certain date, and also the exact dates when we applied the treatments were there as well. So, but here is probably more important, how we actually collected the data. So within each replication, we use 4 trees, as I mentioned, right, per treatment. And we randomly pick 4 branches per coffee tree and 2 branches per side. And we tag those branches, and we essentially take 2 measurements on each of those coffee trees. So first measurement is, we call it incidence of the disease. Essentially, we on those 4 tagged branches, right, we counted all the leaves, how many number of leaves, what's the total number of leaves on each branch. And also we counted how many of those leaves are infected with coffee leaf rust, the fungus, and then we divided that. That is that is called the incidence of disease. That's a standard measurement for not only the coffee leaf rust, but a lot of fungal pathogen disease research projects like similar to this. And then we also had another measurement, which is the severity of the infestation. So that's, by definition, we refer to other literature where from South America, from other places where they had dealt with the coffee leaf rust before. So we kind of refer to their measurements in a way that we can also kind of compare. It's not necessarily a perfect comparison, but we could possibly compare our treatment results to other regions where they had the research on coffee leaf rust, as long as we use similar rating system. So we have this severity of the infection, 0 to 5 scale. 0 means no infestation at all, and 1 means very small, and then as you can see, 2 means up to 5%, 3 means up to 20%, and then you go up. And then 5 means over 50%. Really, it doesn't matter. 50, 60, 70. It's gonna kill the leaves essentially. So that's how we took the data, and we took the data every month after we did the initial application, and we also observe any phytotoxicity effects from any of those treatments. The good news is that we haven't observed any phytotoxicity from any of these treatments for the last 7 or 8 months since we started the research. Okay, so I basically just provide 2 key results graphs here. And the first one is the coffee leaf rust incidence, as I mentioned, the first variable we measure. So essentially, we are looking at the percentage, how many leaves, what's a percentage of infected leaves on those randomly tagged branches. And as you can see here, we have clearly a good candidate in terms of control the coffee leaf rust, which is Abound, which is one of the chemical systemic fungicide. And several other things I want to mention is, first of all, Abound is still holding very strongly after 7 months post the initial application. And my student Lily will continue going to Kona to the trial site for at least twice in March and also in May. We want to see how long the treatment effect can last. And another several things I want to mention is several, we are all interested in knowing if any biological control fungicides can be used. In this case, we included 2, Regalia and Howler. So both of them gave some treatment control efficacies, but only for several months after the initial applications, as you can see from the graph. And they really, the effect didn't last as long as the systemic fungicides, chemical ones. And then another thing I want to mention is, for the control, and you can see the incidence kind of went high and kind of dropped down. It's not because of the trees are becoming better quality. It's actually because of too many leaves dropped on those controlled trees. So there's not much, calculation we can run. So that would be the main reason why you see this drop. It's not because like I mentioned again, it's not because the trees are getting better in terms of condition. It's because too many leaves dropped on those untreated control trees. So this is the second variable we measured. And again, as we can see, the Abound was clearly the best treatment among all the treatment we tested. So if you recall that 1 means only up to 1% of damage. And so this is still holding very good, very strong. And for other treatments, they were all able to keep the leaf rust severity level under 2. That means 5% less of the leaves are showing on the leaf surface are showing the rust symptoms. So which is very good still, but we have clearly 1 candidate of the treatment. It is working very well. compared to others, and also compared to the control as well. So that's a quick summary of the coffee leaf rust research we have done over the last year. And like I said, Lily will continue going to Kona and to keep monitoring the trial. And we will essentially after until May, we will have the extended graphs of both of these measurements that I will provide an update in future webinars or seminars later. Okay, and like I said, we will also repeat these treatments one more time in the second year. So the purpose is to generate more reliable result, I guess, in a way that so we can provide solid or very convincing recommendation in terms of the coffee leaf rust control. So like I mentioned, we Julie, James, Ning, and also myself, we actually went to Puerto Rico to attend the National R4 Education Conference just a couple weeks ago, February 7 to 9th, just earlier this month. And that was held in Puerto Rico. And we actually had a similar R4 trials between Hawaii and Puerto Rico because essentially we grow a lot of crops out of similar crops at both locations. So over there, we did several tours with several different research stations of University of Puerto Rico. And there are two things really we don't want in terms of coffee for coffee industry. One is probably the worst I can imagine is the coffee berry anthracnose. We do have anthracnose that can attack leaves of coffee here in Hawaii, but we don't have this one yet. So this one is just a ridiculously bad looking, and the trees are still on the ground. If the top right picture, you can see actually I can try to point here, all the anthracnose fungus are growing on the cherry, on the berries, actually very extremely bad situation. And this is the display they showed us at one of the arranged field tours, and all these black ones actually are coffee berries. Can you imagine, the fungus are just covering the entire berries. So it's just an extremely bad situation, almost like those post-harvest, fungus growing on the produce, on different produces, kind of things like that. Very bad. And another thing is the insect. It's the coffee leaf miner. This is a moth species, and we don't have it here in Hawaii, which is good so far. And it is considered much worse compared to CBB, the coffee berry borer, in terms of insect problems in Puerto Rico. A very severe infestation over there as well. They can block the nutrient delivery, the water delivery, and also the reduce the photosynthesis capacity when you see leaves are damaged like this. So this definitely we don't want them in Hawaii, but nobody can guarantee they don't come here one way or another. So we just want to be sure that we are aware of this and somehow get ready for this new pest that we don't have here in Hawaii yet. All right, I want to acknowledge all my lab members, former lab members and current lab members who contribute to a lot of research. That did not necessarily all contribute to the coffee leaf rust research, but we work as a team overall. And I always have this disclaimer, any mention of any products in active ingredients is only for the research purpose, for information only. And it's not necessarily saying that okay, we are only recommending the certain products over other products, those kind of things. And we are not basically standard disclaimer I always have on any extension talks like this. All right, in the last slide is the acknowledgement slide. I would like to acknowledge all the funding agencies. For this one, particularly is the USDA NIFA and coming through the the SHAC, the organization for the the funding to UH to my program. And I would like to acknowledge James and also my other lab members and other colleagues, and also Kona Hills to host us for this for both the SCI trial and our R4 trial. And we are gonna conduct the second year and new R4 trial at their location again later this year. All right, so that's all I want to share, and I can take any questions or any comments